Very, very, very big welcome to all of you to the very first 2024 Snow Leopard Network webinar. This is an exciting one indeed. My name is Sibylla Norris. For those of you who don't know me, I was on the Snow Leopard Network Steering Committee back in 2012 to 2018, and it's very much my pleasure to welcome you today. Our speaker, Dr. Carol Esson. I'm so pleased to welcome you, Carol, as I am on one end of the great Australian continent and you are on the other. Welcome.
Thank you, Sibyl.
Carol.
I'm very happy to be here.
Wonderful. So I'm intrigued by the title of your talk. What's behind a snow leopard spots? That's something that a lot of people would be asking, I'm sure. So a One Health approach to investigate emerging disease in the iconic snow leopard. Carol will present us today the key questions and outcomes of her PhD research project on snow leopards in Mongolia's Gobi Desert. Carol graduated from the University of Sydney with an honours degree in zoology, and she has worked on numerous iconic Australian wildlife species, including koala, wallabies, and the quoll, which is one of the more unusual ones. Carol has also completed a veterinary science degree and has worked and continues to work indeed in veterinary clinics. Her work with snow leopards was sparked in 2011 when she volunteered with the Snow Leopard Trust in an ecological study in the Gobi Desert. She decided there was an urgent need for a disease study involving snow leopards and began a pilot study, which later developed into the basis of her PhD. Welcome, Carol. Welcome, everybody. Please, as we go along, if you have questions, pop them into the chat bar and we will be giving Carol an opportunity to discuss those after her presentation. Thank you very much, Carol.
Okay, thanks, Sibyl, for that and welcoming speech, and thanks, Raki, too, for asking me to present this webinar, which I was very pleased to do, because it's nice to be able to revisit my PhD work, because it was actually a lot of fun most of the time. So rather than keep everyone waiting, as Sibyl said, my talk is going to be looking at my PhD work and looking at utilising a One Health approach to investigate the emerging diseases in snow leopards. Okay, now for those of you who aren't familiar, which is probably not that many of you with snow leopards, I was just going to give you a little bit of background information. The snow leopard, as you probably all know, is an endangered apex predator. There's approximately only 3,500 to 7,000 wild snow leopards remaining, which is not a very large number at all. And these are distributed over 2 million square kilometres, crossing 12 countries in the mountains of Central Asia. No, we're doing it again. Okay. Now, current threats to the snow leopard include poaching, as in for this skin, but there's also poaching for traditional Chinese medicine, and that uses normally about 200 to 450 animals are killed each year for this. There is also, however, unfortunately, retribution killing by herders for killing of livestock, which I guess you can't really blame them for. So one of the things we also wanted to look at was ways to be able to mitigate this and provide the herders with alternative methods of protecting their goats. Continuing with threats to the snow leopards, in Mongolia, it's a country very, very rich in ore. So there's fragmentation of habitat occurring because of mining as well, even in the Gobi Desert. The fragmentation of habitats also leading to loss of native prey species such as the ibex. There's also a lack of protection, even in the national parks that have been set up to help protect these animals. There's not a lot of policing, so there's still poaching happening there. And then there was and still is the unknown question of disease. Emerging infectious diseases are a growing concern globally, and 70% are reported to occur from wildlife. As we've seen in recent years, a lot of these are zoonotic diseases, which means they can pass from animals to people. And these include such viruses as the Nipah, Hendra, and SARS virus. Now, in 2011, 4 dead snow leopards were found in the South Gobi Desert of Mongolia by the Snow Leopard Trust. And this raised the question of, could infectious disease be an additional threat? Because health of this species in the wild is largely unknown, and there's really only reports from captive animals. So this was basically what prompted the beginning of my research. So emerging diseases and conservation. So worldwide, zoonoses are as I said, are one of the major sources of emerging diseases, and 71.8% of these originate in wildlife, so as opposed to domestic animals or other animals. And as well as these emerging zoonotic diseases, there's always the threat of previously known zoonoses recurring, depending on the environmental conditions. And as you can see in these areas of yellow, that's where there's the highest proportion of zoonotic diseases at the moment. Now, proposed causes for emerging diseases. Of course, there's climate change, which is causing the ecosystems to change. There's the movement of pathogens in the transmission of disease. And with movement of people, as there's more movement between countries, they also move pathogens along with them. With the importing and exporting of animals, this also moves pathogens between places where they were previously weren't there. And also, along with people moving, is the vector movement. People can also transport vectors such as mosquitoes and ticks when they're traveling. The changing in the climate can also change the environment that makes it more suitable for some of these insects to live in areas where they previously haven't lived. So in my study, I was using a One Health approach aimed to identify zoonotic pathogens that may impact the endangered snow leopard. To determine if these zoonotic pathogens were found, if they also occurred in sympatric wildlife, domestic livestock, and humans in the remote rural area of Mongolia in which we were working. 3 was to look at links between these pathogens and the health of the species in the region. 4 was to see if there was any overlap and transmission pathways for the movement of these pathogens between species. And 5, the other thing I wanted to do was see what the perceptions and attitudes of the local people were to the wildlife and domestic animals and the human health. So with this, we devised disease management questionnaires so that people could answer. And then based on the results of these sampling and aims, we were going to formulate management plans and intervention sites to lead to improved conditions for everyone. Now, a One Health perspective was chosen to look at this because snow leopard are really quite endangered, and a lot of scientific research is a numbers game. We knew we probably wouldn't get a lot of snow leopards to be able to get statistically significant outcomes. And as it's what the One Health looks at is looking at the environment as a whole. So you're not just looking at one area, but looking how each area overlaps and contributes to either the existence or the non-existence of certain areas. So you have the environment itself, such as your temperature, your rainfall. So with the One Health, we're looking at the interaction of the environmental components, which of course overlaps with our domestic animals and wild animals and with people. So you can see from this diagram the overlap, because these aren't individual components, but one relies on the other. Now, the study area is situated in the Tos Mountains in the South Gobi Desert of Mongolia, and it's 1,700 kilometres square. So for those of you who hopefully can see that the map, it's the little red dot right there down near the Chinese border. Now, when we were trying to work out with the study what animals that we should include and what samples we should take, and how following the One Health approach, what we decided with, of course, having the snow leopard as a central piece, we looked at the ibex. It's one of the wild animals. We looked at rodents, as it's another one of the wild animals that inhabits the area, and there's ubiquitous over the area with several different species. The main farming occupation in the region is goat herding for cashmere and meat. The other domestic animal we looked at was dogs, because they're around the goats. They also interact with the people, and they interact with other wildlife as well. We included water. Oops, I've got that. We included water because all animals come to the water to drink, obviously. So that's one of the major environmental areas that we looked at. And of course, people, because they interact with the domestic animals and the wildlife, either purposely or not purposely. Anyway, the primary pathogens we decided to try, because there's so many to look for, we had to select ones that we could test for. It was just impossible to test for everything. So we were going to concentrate on zoonotic pathogens and ones that have been recorded in other areas of Mongolia and that could impact people, wildlife, and domestic animals in a way that would either make them sick or would compromise their ability to farm and to decrease income. So with the bacteria, we were looking at Yersinia pestis, which is plague, which is carried by rodents. Coxiella burnetii, which is a Q fever, mainly carried by cloven-hooved animals. Leptospira, which again is mainly carried by rodents, but can inhabit any warm-blooded animal. And Brucella, which again is another cloven-hooved animal bacteria. It can occur, of course, in other animals. Viruses, we were looking at canine distemper virus, which of course is mainly in dogs, as the name suggests, but there has been species switching, such as the lions of the Serengeti have become infected with canine distemper virus, and it has caused problems in their population. So we wanted to see if there was any in the area and what impact that, if any, that was having on the snow leopards. Hunter virus is a rodent virus, and it can be quite a deadly virus, and it mainly affects humans. It doesn't normally affect the rodent. Tick-borne encephalitis, because there are a lot of parasites in the area, especially on the rodents. The rodents carry fleas, the goats carry ticks. So the other things we were looking at were insect vectors as well that could carry some type of disease. Parasites, we're looking at toxoplasma, which is, again, rodents can be the main one in the life cycle of the protozoa, and it can cross to other species as well, including humans. Giardia and Cryptosporidia are quite often found in waterways and can contaminate waterways and causing illness. And Echinococcus, which is a tapeworm, and that can cause issues both in meat and in people. Okay, so the samples that I collected from each of the animals was blood, feces, and external parasites. So that was from the snow leopards, rodents, dogs, and goats. With the ibex, because it was very difficult to catch them and it was just too time consuming and out of the scope of this project. So we collected feces from the ibex because it was easy to see where they'd been. We could get fresh samples. Plus, we could always test the feces for bacteria using next generation sequencing. Okay, so the plan when we were catching the animals was to do basic health line monitoring of each animal we caught as well, to assess what their health status was, whether they were sick, healthy, before we started. So each animal was caught, was given a basic health profile, and it didn't matter if it was the snow leopard or a 20-gram. So for each of these animals, we did basic monitoring, including heart rate, body weight, temperature, body length, body condition, foot length, ear length in the case of the rodents. So starting with the snow leopards, how do we catch them? This is a snow leopard. For those of you who haven't had the privilege to be able to go on one of these trips, this is a snow leopard snare. This is Orion Johansson. He was at the time a Swedish PhD student, also doing his PhD on the ecology of the snow leopards. And he caught all the snow leopards in the study. So he'd already been working on this for several years and had it, and was collecting data on home ranges, fecundity, kill sites, etc. So we joined together to utilise resources. When the animals were darted, or trapped in the, when the animals were trapped in the snare, it went off, and this antenna was hooked up to each of these snares so that an alarm went off in the ger where we were staying, so that we knew which snare went off and when, so that no animal was in the snare for any longer than 20 minutes, day or night. So to start with, of course, the animals were darted. They were checked. They had radio collars fitted, so that these were both GPS and VHF radio collars, so they could be monitored two ways. We had pulse oximeters, so we could measure their pulse and oxygen sats. Plus, I'm a bit of an old-fashioned vet, and I always like to use a stethoscope as well, because I never rely on machines that go bing, because they can fail. At the end, we would weigh the animals. We would also, it was too hard to put in all pictures, but we'd also measure them, measure tail length, take paw prints, look at their teeth, collect any external parasites if we could, and then give them a reversing agent, and they would wake up. This was one of the few animals, Natai. We called him Natai because I think it can correct me, Byra, if I'm wrong. I think it means sunny in Mongolian. And he was, I think, one of the only animals we actually caught during the day. The rest was usually at night when it was freezing, but he didn't really care. So he was a magnificent, magnificent boy. Then we moved on to the rodents. Rodents we used as a sentinel species because they were literally everywhere over the study site, and numerous species as well. So we had traps. I don't know if you can see it in this photo. There's a little orange blob here and then another one up there. So this was my way of working out where I put my traps. So there were different regions that we trapped in. This was the mountainous rocky ones where we caught these little I guess they were like a mouse or like a dwarf hamster. These were on the plains, which was very hard to disguise. The traps, we sort of had a few competitions about who could build the best trap disguise. And that was the gerbils or the red-cheeked ground squirrels were there. We called these the red bushes. They don't look very red in this picture, but they were sort of in the gullies and they had roots underneath them, which these gerbils would have their burrows under. So you could tell which species after a while, not to start with because no one had actually looked. You could see where these rodents would be and who you would catch where. Okay, now some of these rodents were, despite their size, were very feisty little guys. We did try sedating them, but it was because they were so little, it was really unpredictable, and sometimes they just slept for ages and ages. So we came, and this is one of the things of working in the field, you become very ingenious and devise methods that you probably wouldn't do anywhere else in a lab. So I think this might have been an empty toilet roll holder with gaffer tape to act as our straitjacket we nicknamed these guys the lion mice because they could really bite when they wanted to. So when we wanted to measure head length, tail length, foot length, we could sort of keep it restrained without damaging it and be able to do all those measurements as well. It would take two of us to take blood out of these little rodents, but you could manage to get it eventually. This was a solar-powered centrifuge. So this is the outside of the gear, and these panels are solar panels. And it used to be enough to power this little centrifuge, which I would put my blood samples in to spin. I just like that photo of the ibex. I thought it was really nice. So because he was the natural food source of the snow leopards was one of the reasons we did want to include them, and how they could run up and down these mountains was beyond belief, and how they didn't fall, I don't know. Now, when we started investigating the domestic animals, so between 2012 and 2015, I sampled 270 goats and 41 dogs. Unfortunately, dogs just went to the toilet wherever. We didn't really collect any fecal samples from dogs. But I did, which the herders thought was a bit gross, I would put a little swab in the rectum after we handled them, and took a little bit of a poo sample if I could get one, and put that in ethanol to keep. And again, this doesn't show particularly well. These dogs, most of them weren't pets. They were there to act as a guard for the goats. So they weren't used to being handled. And quite often the owners were scared to touch them as well. So I did have to quite often muzzle the dogs. I didn't sedate them because people wanted them, they didn't want them sedated. So we had to go with what they wanted. So I would get blood with a couple of people holding the poor dogs down. Some were okay, some would let me do it, and without too much fighting. The goats were pretty good on the whole. For one of my ethics committees, I had put in, they had asked why I hadn't put in the sizes of paddocks that the goats were kept in. So I sort of had to explain to them that, and they wanted to know the methods of how the goats were caught. And it's like, well, it's the Gobi Desert, there aren't any fences or paddocks. And most of the herders could go up and catch the goats by hand, so which is what they did. The main problem with taking blood was it was so windy that the hair would just blow in the way constantly. And again, because they were goats for cashmere, I didn't really want to shave any area. With the herd health in the Gobi Desert, when we were there, there were no current vaccination programs. There had been when it was under Russian rule. I think that stopped in the 1990s. But there were no reports of disease studies previously in the area in the times when they were vaccinating. So it was really hard to know what was present and what wasn't present in the goats during those times. There's been some reports in other parts of Mongolia with camels, not a lot on goats. So this guy was very curious. He'd sort of come up all the time. There was overlap between these two, as you can imagine, although the ibex tended to go up higher than what the goats did. And generally when the goats were taken out through the day, the herder would go out and keep the goats contained within the flock and take them to areas where they were to graze. So the average goat size, average goat herd size was about 250 animals. So I'd sample about 5% of each herd, and that was a mixture of males, females, young and old. So it was just sort of a random sampling. And I also collected fecal samples, of which there were numerous. So as you can see, a picture of the puppies, which I did them as well. This isn't stones that these puppies are sitting on. This is goat feces. So there was a large opportunity for there to be an exchange of pathogens from the feces to the puppies and to the people too, because people would be walking among this, and because it was so dry and windy, it'd be constantly stirring up dust. The dust was incredible, and so any airborne particles you would be breathing in all the time. This was with the dog sampling. This one was a friendly one. Gustav managed to make friends with this little dog. It sort of followed him for quite a while. It didn't want to go back home. So Gustav was the other senior research officer from Snow Leopard Trust. So now the human health in the Gobi Desert. So I went back through the literature, but there were very few reported studies on zoonoses in this area of Mongolia. It's a little remote area which sort of had been not had a great deal of attention paid to it. In Mongolia as a whole, there had been reports of rabies, plague, tick-borne encephalitis, Lyme disease, which is again from ticks, rickettsia, which again is usually from ticks, malaria, which is from mosquitoes, echinococcus, and brucellosis. I didn't test for rabies because I thought it was too dangerous, plus anything that had rabies would have died, and I probably wouldn't have seen it anyway because it's a fatal disease. Same with anthrax. Everything dies, so the chances of actually sampling something and getting a positive for that was very minimal. Now, with the herders in the area, there was approximately 90 families within the study area, and they are nomadic people, and they do tend to move, a lot of them, with the seasons because it gets very, very hot in summer and extremely cold in winter. The surveys we distributed to about 45 families. Well, I had my interpreter and he would explain the surveys to the people and what we were after. So what I asked was about the health of the families, if anybody was sick, what their symptoms were, the health of their stock, and again, what their symptoms were, and if there was any wildlife they may have observed that have been sick or killed or what have you. The local doctor and vet who are in the town 50 kilometres away were also given the same surveys to complete. Now, this is just a map showing where the water holes were distributed over the study site, because I wanted to get a distribution of water holes from the more mountainous areas to the more plateau areas, and to try and get a cover of the whole area. A lot of these water holes, Orion had camera traps on as well, so that we could see what other animals, besides the animals in our study, were visiting these water holes. So in sampling the water holes, I sampled 11 water holes and wells over the area. Now, this is one of the wells. So this is a cover for the well. There's usually a bucket that goes down into the well, and water's poured in here for the goats. There were a few camels, and there were also some horses for the stock to drink. And also people drew water from these wells for their consumption as well. So as you can see, it's not a very closed system. So a lot of dirt, a lot of fecal matter, a lot of bird droppings and insects, bugs could also, and also sometimes rodents, so dead animals would quite often be in these as well. This is one of the natural waterways. This was not long after the snow and ice had thawed, so there was water. It was actually quite salty a lot of the time, the water. So I sampled from both the wells and the natural water. Now, what I did, this is called a Sterivex filter. So I utilised a sterile syringe, and inside this little filter, there is a cellulose, well, this was a container which contained a little cellulose filter, which read down to 15 microns, 25 microns. So I would push 100 mils of water through this filter, and then I could undo the top of it and then take the cellulose filter out, which had hopefully trapped any particles going through it, put that into a sterile cryo vial, and take that back to the gear awaiting transport back to the lab. I put this one in just because I liked it. This little girl followed us around everywhere, and she also really loved her little twin goats, which slept with her in the gear as well. So there was very close communication between the pets, or well, not really pets, between the animals and themselves. Now, one of the difficulties I had being in such a remote area was in the storage of my samples, so that they would be suitable for testing once I got them back to the lab, because there was very limited ability to do any tests out in the field. There was no electricity. There were no deep freezers for -80 sample storage. It was really difficult to try and get liquid nitrogen canisters out there, or dry ice out there for storage, because it was like a 20-hour drive, or 20-hour drive from Ulaanbaatar, the capital, or an 8-hour drive from Dalanzagad, the closest town that a plane would go into. So I was lucky enough, this machine on the left is called an Ibaxis iStat machine, and it's a handheld battery-operated hematology and biochemistry analyzer, which runs on very, very small amounts of blood, 2 drops. So I could, in the field, analyze blood straight away. These next Advantech, these are called Nobuto strips, and these are like a filter paper. They're just like thin strips. I should have actually taken a photo with some of them out so you could see, but they're like a thin piece of filter strip which hold 400 microliters of blood. So what you do is coat both sides of the strip with blood, let it air dry, and then it's stable. So you don't need refrigeration. It's stable at room temperature, as long as you keep it dry, which we didn't have a problem keeping it dry there because it was very dry. With these, once I got back to the lab, I could elute the blood from the Nobuto strips. And from that, I could run sera and run the tests on the blood as if it was fresh blood. Before I went, I actually did a test with some fresh dog blood and dog blood that I had stored on Nobuto strips to see if there was any difference, and I couldn't find any difference in the results I got from that. This next one, the FDA Classic card, the Whatman FDA Classic card, is another way of storing blood. It's again a bit like a filter paper, but you can get DNA from it. So if we wanted to do genetic testing or look for genetic for the blood smears to see what the bacteria was, or the DNA of the snow leopards or the rodents, we used the FDA cards because again they only used 1 drop of blood. And my centrifuge, I explained that in the previous slide. So the lab work that I could go in, the lab work I could do in the GARE was very, I was given a little bit of this table to work on. This is on his computer. This was my little spot for my centrifuge. So when it was too windy outside to do work, I would bring samples inside, I would spin the blood and then take off the serum, put it in cryovials. Now we had a fridge that we could put some samples in, but it only went down to -4. So I wasn't sure how well the serum would keep for a month at that temperatures, but I took it anyway in case. And this was just a bigger photo of the iStat machine. They actually have a new one out now, slightly different format, but you can see that it gives you sodium, potassium, chloride, calcium, and carbon dioxide in the sample. So you had several different cartridges which measured different parameters. Then when I took the samples back, I analysed my samples in Uppsala in Sweden. I used ELISAs to detect for primary exposure to pathogens on the serum that I eluted from the Nobuto strips. I used, for the leptospirosis, I used MAT to distinguish between different serovars. For those of you who don't know about leptospirosis, there are many different varieties, I guess you'd say, and it was good to be able to see which varieties occurred the most prevalent in Mongolia. I also used I also used PCR to determine the presence of pathogens, and then the water samples and fecal samples I sent to Johan Michaud in Belgium, who did next generation sequencing. That was just too out of my depth to be able to perform that sort of, and he had all the equipment for running those sort of tests. So what did we find? So this is just from between 2009 and 2015, 20 snow leopards were sampled. So this is just table, as you can see, is just giving an indication of the average weights. So males were roughly around 43 kilos, females 36 kilos. So there was a sexual dimorphism between the males and females. Heart rate, body temperature, and respiration rate, there was really no significant difference. And the subadult males were also significantly bigger than the subadult females. Now, the results, I'm sorry, I'm doing this, it's a little bit condensed, but to fit into the time, I sort of had to squash anything down. So please feel free to ask any questions at the end if I've missed something that you'd like explained further. But making it look pretty with this graph so we could identify. So the snow leopards are pink, the rodents are green, the goats are yellow, and the dogs are red. So just to remind you, the pathogens that we were testing for and that we got positive results were Coxiella burnetii, which is Q fever, which is a zoonosis for people, and it presents as like colds and flus. Interestingly, it was the highest in the snow leopards. Quite often Coxiella is in afterbirth and things like that. So that was an interesting result. So I would have thought the goats would have been higher than that. In toxoplasma, again, interesting result with 90% being in goats, with dogs 66%, and rodents, which I thought would have had a higher percentage, but they are an intermediate host. So the dogs and the goats had the highest percentage. And it is a concern that they were highest in the goats because people had more of an interaction with the goats during slaughter, during combing, hand raising them. So it's usually transmitted by a fecal oral route. That normally your cat is the definitive host. If we were looking at a domestic animal cycle, your normal pussy is the definitive host, with the rodent being the intermediate host for the parasite to mature in. So whether the goat was taking the place here, it was an interesting phenomenon. Next, leptospirosis. As would be expected, it was highest in the rodents, but there was a significant amount found in the snow leopards as well. And interestingly, the cerevar that was found in the snow leopards was Leptospira australis. So I was actually accused of planting some virus in there, but I honestly didn't. It was just fortuitous, and that hadn't been ever identified in Mongolia before. So that was a first. All these are a first for what we found occurring in that region of the Gobi Desert. And only 5% of the dogs had leptospirosis, so which is a good thing because it's highly fatal to dogs. I don't know about snow leopards because it hasn't actually been identified in them before. Rodents are usually carriers. It's a bacteria that resides in the kidney tubules. So whenever the rodent is eaten or urinates on something, then the bacteria is shed in the urine, which is why it's a problem when they're found inside people's gears. And then only 2% of the rodents had hantavirus, and I think it was the pulmonary hantavirus, which again, it was the first to be found in that region. It is a zoonotic, and it can be quite fatal or make people incredibly sick. Now, when we look across our study area as a whole, looking at the three, I didn't look at the hantavirus here because there was only, I think there was one rodent in the end. You can see that the pathogens were spread over most of the study area that we sampled. So these were present everywhere. Interestingly, brucella wasn't identified in the goats because it's quite prevalent in the rest of Mongolia, and there's quite a few studies have been done where it's positive in camels. Dogs were negative for canine distemper virus, which was good. It could possibly be because this is such an isolated area that there isn't any new animals coming into the area, so that these dogs may not have ever been exposed to the virus. So that would be interesting to see. Yeah, and the pulmonary hantavirus was present in the rodents. Now, the ticks, thank you, Gustav. That was his photo. He said if I ever gave a presentation. So that was a really nice photo of a tick. There was also another tick, which I unfortunately didn't get a photo of. It was a big stripy one, and people were actually quite scared of it because it said they said it made them very, very sick. And I didn't really get one to test, unfortunately. So it would have been, and I couldn't really work out in what way it made them sick. So that would be an interesting one to follow up on. And just because no vet talk can be complete without it, there's a snow leopard poo for people to have a look at. So ticks and feces were found to carry several potential zoonotic genera of bacteria. These included Bacillus, Bordetella, Coxiella, Rickettsia, and Yersinia. Yersinia pestis is plague, but it wasn't pestis that we found. However, they can all cause significant zoonotic infections. Now, the water sampling, again, was quite interesting because this is frozen, if that's not obvious. This is usually when this melts, this is usually like a little river running down here. But when it's really, really cold, the water freezes. So I got a sample from a water hole up here where we smashed through the ice and got a sample from there. And in contrast, this was down in one of the areas where seen birds drinking, the goats have been up there. Obviously, there was footprints all around the edges where animals had been drinking, and yet no significant zoonotic pathogens that we tested for were detected in these water. So there was no Cryptococcus, there was no Giardia, which I found really interesting. I thought there would be something there. So it's probably worth retesting the water, and especially now this is that new techniques coming out. Probably eDNA would be a better way to go now to check the water samples to see what else is in that water. Now, the results of the surveys that we gave the herders, and it wasn't just the male herders, they sat down and quite often did it as a family. 60% of the surveys were returned, which was quite a good percentage. There were no reports of any recent epidemics. The most common illness reported were colds and flus, which, because of the nature of the environment, it was just, you'd go out through the day and it wasn't, oh, is it raining today? It would be, is it windy today? It was just windy constantly. And we always had what we called the Gobi drip, where your nose just constantly dribbled because of the wind, the cold wind blowing it on all the time. So colds and flus, because of the weathers, may not be that surprising, but 6.3% of the goats and 10.5% of the dogs showed prior exposure to Coxiella burnetii, which is otherwise known as Q fever, which the symptoms of Q fever are cold and flu-like symptoms. It would be interesting to be able to see what the people were tested for, but they very rarely went into town to go to the doctor, especially for a cold and flu. So that's sort of like another whole project in itself. Now, interestingly, the doctor reported that brucella, hepatitis, and blood pressure were the most common ailments of people, which you can understand. Well, the brucella was confusing because it was like, okay, we didn't find it in any of the animals we tested. So that, again, needs further investigation. Hepatitis and blood pressure, well, there was a reasonable amount of vodka around. Whether that contributed to that, I don't know. But yeah, they were very happy to share it with you on occasion. According to the doctor, all the children were immunised. According to the surveys, only 48% of people were immunised. So it might be that all the children that went into the doctors got immunised, but there were ones that they didn't know about that weren't. And because the Mongolian people were very private people, and, you know, I was an outsider coming in and asking them I guess, reasonably personal questions. A lot of them that I didn't see a lot of the time were more reticent, even though I had either Nadia or Sumbe with me to ask. They were reticent to say if they'd been sick. That was personal to them. So there was probably a bit of bias in some of these surveys as well. Okay, so when we checked with their most common cause of death, most of the predation they said was by wolves. So that was interesting, 70% as opposed to 50% by snow leopards. We did see one wolf in the area which had 3 legs. Obviously, it had lost 1 leg in a trap. Other causes of stock death included 13% due to mortality, giving birth, which again could be due to brucella, but even though we didn't find it when we tested. 24% had abortions, 4% due to eating poisonous plants, and 13% due to unknown reasons. Only 5% of the herders treated their stock for parasites, and 3 reports of illness were observed in wildlife, and that included rabies in 1 fox. So it's such a desolate area, and carcasses don't last long, so it could be animals died and weren't even noticed. Okay, so summarising what we've found here, Toxoplasma, Leptospira and Coxiella were identified in moderate to high prevalence with previous exposure in all the animals we tested. So that was a significant finding. And this led to ways of transmission paths that we could look at. And this looks a little bit jumbled, and we're looking at it from a One Health perspective, but we can see where the environment, the people, the wild animals, the domestic animals, and the rodents, where there are numerous opportunities for pathogens to be transmitted and can be transmitted between all these components of the study, especially when you consider quite often the rodents were inside the gears. People were killing the snow leopards, so being exposed to carcasses that way. They were killing combing the goats or killing them for meat and purposes like that. So again, they were exposed to pathogens and parasites that way, and the dogs as well, because they scavenge on the carcasses. So it's not a simple entity to look at. It's a combination of all these pathways that have to be taken into account. And one of the things that we wanted to look at was where we could interrupt these pathways to minimize disease transmission between the animals, to stop things like an epidemic happening in the area. So I think I might, seeing though time's limited, I might skip those ones. So these were some of the issues that I came up with that we could instigate to try and mitigate some of these pathogen risks. And that would be vaccinating the goats if possible, but again, these things cost money. Treating the dogs and the goats for internal and external parasites, which would help control that transmission. This would decrease possible transfer of Q fever to the snow leopards by decreasing the infectivity of the goats. It would also decrease the contamination of the pasture with parasites and pathogens for competitive grazing, like with the ungulates. There were also bighorn sheep there. This would decrease the ticks as potential vectors for carrying disease, or pathogens rather. This would have flow-on effects that we wanted to get across to the herders, that it would increase herd productivity. If we could increase the health of the herds, then they would get a greater income from the herds if they were in better health. And it also might help with mitigating herd or wildlife conflict if they were getting a better outcome, like increasing the number of births from their goats. They hadn't actually thought that they might be losing kids due to disease, they were assuming they were losing it due to being hunted. So that was one of the things we were trying to establish. And it also would reduce the risk of human illness, which then they wouldn't be off work. And again, it would increase productivity and for the family as a whole. Okay, this is just some other things that we thought we could do. Rodent control inside the gears, encouraging people to wear gloves and face masks during the goat slaughtering, aiding in the birth and combing the cashmere, just because of being so dusty, and aerosolizing everything. So for future work, I think we need to continue pathogen monitoring and surveillance of the snow leopards to build on this baseline knowledge that we've gained, and to look at a greater insight into the transmission of the pathways of these pathogens and using molecular epidemiology as the next step. Refining the methods for collecting and storing the goods in remote regions, then we can increase the accuracy of our results. And increasing the rapport and capacity building with the local people, if they can see that they're not only are we there to help preserve the snow leopard, but also to help preserve their lifestyles and income. So that was the main part of my talk. So due to the endangered and hence limited numbers of snow leopards available for sampling, by utilising a holistic approach such as the One Health approach to detect pathogens, did lead to a greater understanding of the snow leopards and the whole environment in which they lived. It showed that there was a risk across hosts, that is the wildlife biota, the domestic animals and the people, for the pathogens to move among due to their ecological connection, therefore increasing the possibility of pathogen spread between species and the environment. These are just some of the people that I wanted to acknowledge for all the help I got during my PhD, my supervisors, Gustav, Charu, Orion, Jonas, or people in Uppsala, people back at JCU, people that helped in the lab in Uppsala. They were just amazing. And as Sybil said, there were a few moments that I just wanted to reflect upon, I guess you'd say, that makes this type of work, for me anyway, to be absolutely amazing. Like one of the most memorable occasions was my very first snow leopard capture. This was Aztai. I think he probably resented Orion. I think he'd been captured numerous times, but for me, this was the first. So this was fantastic. It was midnight. It was freezing. I could hardly feel my hands, but I wouldn't have swapped places with David Attenborough at that point in time. It was just amazing. There were also, as with any field work, times when you felt like giving up, like when you had just put out 80 traps and hiked up and down carrying traps, and then it snowed, and then you had to hike back up and down, bringing them all back in again. There was also the weather to deal with. This was a road, which then turned into a flash flood. So our trusty Russian van became the Russian aqueduct. And then there was the cleanup after the flash flood, which came in and went right through everything in the Gare, which you wouldn't think so looking at the dry ground there. So I'm just rushing through these to get this in. And also the traps suffered again, had to go out and collect all the traps, make sure that no animals had got caught in them or had been drowned. This is our Russian van, and as you can see from the skylight at the back, it was a very ominous day when the flash flood came through. But this machine just went everywhere. It was invincible. Sometimes you trapped animals that you hadn't thought you would trap. So then the next problem was getting them out of the trap. So especially if they weren't very cooperative. This was home. This was the Gare in which we lived for 4 to 5 weeks at a time, very cozy, ranging from 2, 3, and 4 people at a real squash with our beds around so this was the bedroom, this was the office, this was the sink where we washed our hands, and this was our kitchen. So this was life in the Gare. This was Gustav trying to act as a lightning rod during a thunderstorm as we were really desperate to find a cub in the den. And we wanted to do it before the male that we knew was around appeared on the scene. This is just to show what the area was like and how the people lived. They were just amazing. I like where the goats found something to eat. I don't know. There was, as far as I could tell, there was nothing there. And they do live in this really harsh environment and survive. We also had our little bit of fun. This was a cook-off to see who could make the best cake. Mind you, we had no flour, eggs. We did have sugar. This is Gustav. And this was Jeff. He was one of the volunteers from America, Gary. And these were the products of their cook-off. And this was to celebrate Orion's 1000th day in the field. So that was a big milestone. And I think he appreciated the effort that went into making the cakes, although I don't know if they actually tasted that good. And after every celebration, there was always the cleaning up at the end. So when it came to cleaning up the traps. And this is also, sorry, one of my very favorites. This was inside the Gare, looking out. And see this black plastic over here? That was our toilet, which when you had to go out in the dead of night, and as you can see, it was snowing, was probably not the most comfortable place to have to be in the night. So you made sure you went, mind you, it wasn't that comfortable during the day either. But yeah, that one will always stick in my mind as a field toilet. And then last but not least, I'll go through these ones really quickly, but this is the last video I really wanted to show you. We spent days and days radio tracking one of the Anu, one of the females, to a den. And we were trying to pinpoint where that den was and when she was there. So we were pretty sure she was in this den here. We didn't know if she was in there because with her collar, it only read the GPS every 5 hours. And because of the mountainous area, the signal will bounce backwards and forwards for the VHF frequency on a radio collar. So we weren't sure if she was there or not, but she had been sitting still for about 4 or 5 days. So she either had a kill site or she had a cub. So again, using field work, field ingenuity, we rigged up a camera with the mirror of the my mind's gone, the compass on it. We devised a plan, which I will speed this video up, which we were going to go and look into this. Into this hole. This was marching off, going into the hole, making sure we had everything.
Got a bandage.
Again. And then what we did was turn the camera on and stick the camera over. And what we saw that's the rock wall. And on the other side was this. It was just, we couldn't see, we couldn't hear because of the wind, but the look on her face is just like, if anything comes in here, I'm going to kill it. It was just amazing. So sorry, this video is a little bit not unprofessional, but as you can see, we just had stuck the stick over the thing, and so we couldn't actually see what we were looking at. But she was watching that video camera and just keeping quiet. So luckily, they're not a very aggressive cat.
Amazing. What a study. What a study.
Yeah.
The breadth and the depth of what you were covering there. Truly amazing. I'd like to suggest that we go on perhaps another 10, 15 minutes. I am hoping that most people can stay with us because I would like to introduce Dr. Muhammad Ali Nawaz, our discussant today. I'm sure many of you know Ali. He is a legend in snow leopard research and conservation. Quickly, I'll just cover some of his background. He holds a PhD in ecology and natural resource management with 25 years experience in the field of wildlife and conservation. Ali's research focuses on understanding the ecology and the conservation issues of threatened species, and he employs both field and molecular techniques. Ali, would you like to give us some of your impressions, your thoughts of today's amazing presentation? Thank you.
Thank you so much, Sibylla, for a very nice introduction. And thanks to the Carol for this comprehensive presentation. I was, through some initial chats with Gustav and Orion, I was a bit aware of this, important study ongoing in Mongolia. But with the presentation today, I got more comprehensive view of what has been achieved. And this is remarkable. This is remarkable. We are working to address this disease issue in snow leopard habitat in Pakistan for now more than 10 years. And I have been looking into the literature, and I know very well that this is the area which has been kind of poorly addressed, particularly in the snow leopard habitat. So there was much more room and need for this kind of work. And the approach that Carol has adopted, this is, quite comprehensive, I must say. He has taken into account so many different taxonomic groups, both from the wild as well as the domestic animals within the framework of One Health, that approach that is very appealing and very much relevant to the conservation approach that most of the countries are adopting. So I would like to, I don't have much to add, but I would like to, try to link it with the conservation that majority of our community is focusing on. In conservation, we often aim for maintaining ecosystem resilience and integrity. The way what we mean is we look at what is the species composition there and what are the ecological interactions that we try to preserve. But this concept, if we look at that ecological resilience concept in the framework of homeostasis, which essentially deal with regulating the internal system, then we believe the ecological resilience and the, this One Health program, the approach becomes the same. Because without addressing this disease issue, sometimes it can shake the whole ecological interaction and overall the composition in a ecosystem. We cannot achieve much on the conservation as well. Not only from the academic and scientific point of view, but also from the practical conservation perspective. How? We did a bit review of the consequences of these diseases in some of the region. And what we found out, it was quite alarming. We often try to address depredation from the snow leopard, from other carnivores in order to build financial resilience and acceptability in the communities, because they lose generally huge amount of their financial worth from these from the depredations. But in reality, what we found was people, they lose 3 to 5 times more livestock to disease as compared to they lose to the carnivores. So the economic burden on the community is much higher.
Mm.
And though there is not much data still, but we have found there's a widespread of diseases, so many, these brucellosis that she was mentioning, we see rinderpest, foot and mouth disease, and so many other diseases, they are taking toll. And even in some cases, we saw the prevalence of ectoparasites, particularly the sacroptic mange. This has been the major culprit in causing a huge amount of mortality, as well as even not the mortality, they weaken the animal. The overall productivity of the animals goes down. So the farmers are unable to get the required amount of milk and meat and other things they intend to get from. So when see with the disease spread, which is generally there with so many diseases, and which it is coupled with the poor veterinary services generally in the snow leopard habitat, then the economic burden is higher. So the community which is already suffering from huge economic burden, when they lose even a single animal from a snow leopard, then their tolerance is even much more stress. And then convincing them for any conservation work becomes difficult. And Carol mentioned vaccination thing, that we believe it is very important and very effective. We have found we are doing it now for about 10 years in Pakistan. And what we found is when we try to do the comprehensive vaccination program, we found that even 1 year vaccination mortality rate from the disease, it goes down 50%.
Wow.
And even just in 1 year of vaccination in community where it goes around 50%. So you have reduced the economic burden by 50%. And if you continue it for a longer period, we see after 3 years, it goes down from 80% to 100%.
Wow.
Then it becomes very important, conservation tool as well. And I was amazed when I met one, community in Pakistan, northern Pakistan, Parsan community that is living at 3,000-meter elevation. So they adopted a couple of approaches working with the Snow Leopard Foundation. One was, of course, they built predator-proof corrals. They did effective guarding of their livestock. At the same time, they partnered with us and they started the vaccination. So the year I visited them, I found that there was a zero loss of livestock in that community in that particular year. That survey I did in 2010. There was zero loss. So they preserve animals from predation through the corals and active guarding, and then through vaccination, they did not lose any animal to disease. So they cured, all of their important wealth, which is very crucial for the well-being of the community through this multiple approaches. So this, understanding disease and managing disease is very important for well-being of the community. It is important to push the conservation agenda forward. And even beyond that, there are several studies. We see that these diseases, then they pass on to mountain ungulates. We have seen the cases of collapse of markhor, blue sheep, and many other species. Substantial, their population collapse in Tajikistan and Pakistan and other things. And even to snow leopard, we have seen cases, snow leopard suffering from mange. And one of that, even, it came to a village and we were captured, and he was so weak that it could not finally survive. I have seen some brown bears in the Diosai National Park that they were suffering from this ectoparasite, and they were, their skin was falling apart and they were getting weaker. So I believe this is, this is a good start, and I would request Carol to expand on this work, and maybe if the ectoparasites are not part of this study, those could be included, and maybe he may develop a partnership with other range countries to expand this work to the wider range of the snow leopard So these benefits could be shared across a larger range. Thank you so much, Carol.
Thank you for the words, Ali. And yeah, I agree with you with that. With just doing some of those basic health parameters, it increases the productivity of the herds so much, which has a flow-on effect for the whole community, including the wildlife as well. So yeah, definitely important. It was interesting though that most of the rodents had fleas, but it was hard to tell with the dogs because getting close enough to actually look was difficult. Didn't see any on the snow leopards, and we'd go over them and we'd collect as many ticks as we could find, but there wasn't huge numbers. But yeah, there were huge numbers of fleas on the rodents. So, you know, the possibility of plague was always there as well.
Yeah.
Well, thank you so much, Carol, for that amazing presentation. And Ali, for your insights. I think the points that you brought out in your discussion of Carol's presentation are so valid. Economic impact and the importance of this work. And I'm hoping that many people with us today will go forward and be involved in exactly these types of studies. It's been a testament to how interested people are that we have gone over time by about 20 minutes, but nobody has left the session. So everybody is still here. They are absolutely fascinated by this topic. So thank you to both of you again. Carol has very kindly offered, we did have a few questions. Carol has very kindly offered that she would be happy to answer questions through her email, so that rather than going into questions now, please do look out for her email address at the end of this presentation and contact her direct. I'd also like to remind you that Raki has already advertised the next Snow Leopard Network webinar for next month, so that should be in your inboxes. Please all have a look for that and join us if you can. Thank you again, Carol, an amazing, amazing study. And thank you, Ali.
Okay, thanks, Sybil. It was a pleasure.
Thank you.
Bye to everyone. Thank you, Raki.
Thank you all, and bye. See you all soon.